Fine by me.
Okay, you can go now.
Great. All right. So before I start with my talk, I just want to say quickly thank you to Elena and Rani for organizing the event, for having me as part of the event, and also for quickly moving it online so that we can all have this workshop. And I'm looking forward to seeing the other videos as well and the discussions. Regarding myself, I'm a research associate at the Center for Philosophy of Natural and Social Sciences at the London School of Economics. I have a background in biochemistry, but now I'm working in philosophy of biology. Regarding my talk, you might notice that I've shortened my long title a bit. Basically, the case study I'm going to talk about is from pig farming, and it has to do with genetic editing of pigs, and obviously ontology is coming in as well. So just to give the background of my, like where I'm coming from in this talk. The starting point is obviously the paper by Frederick, Rani, and Elena, 2019, and also some previous work, and what they call philosophical biases and how they shape expert-led debates. And the key bias that I'm interested in my talk are ontological assumptions that people make in their arguments or in their projects. These are assumptions about the fundamental nature of the entities or the phenomena they're interested in. So be it the atom, the gene, the organism they're working on. So really the question of what is a gene, what are its key features, what is an atom, and so on. And the problem that Elena and the others and also other people have highlighted is that differences in our worldviews, kind of these basic ontological assumptions, they're like a lens that we use to look out at certain issues, and they can lead to disagreement among scientists and experts, or anyone for that matter. I mean, we're just focusing on scientists and experts here. And that obviously affects stuff such as data interpretation. How do you interpret data? How do you get data in the first place? Or questions of risk assessment. How do you define risk at all? So all these questions can be affected by your ontological background assumptions. And Elena and Frederick have proposed an interesting solution to the problem, I think. I mean, the first point they make is when you deal with these kind of disagreements, you have to, first of all, make your biases explicit so that you know what you're talking about. You have kind of an informed decision. You know where people are coming from. The second bit, and that's the one I'm mainly interested in, is that you kind of then try to choose, you kind of try to judge which ontology is kind of the one we should go for, because they're obviously kind of clashing often, if they are clashing. And so the proposal here is that we should choose the ontology that best matches what kind of current scientific knowledge is telling us about the world. And in a way, that's a proposal that we can kind of resolve disputes between experts by looking at science, kind of look harder, kind of, we really got to figure out what is science telling us. And so this is kind of the thing I'm going to talk about, the issue, the block of issues. And I'm going to do that by looking at genetic engineering, because many people have discussed kind of, well, not many, but there have been quite a few papers that discuss the ontological assumptions that underlie debates about GMOs and genetic engineering. So I think it's a very good case study in general. And what I'm mainly interested in is this point 2, because there is a question that comes up to me, and that is how, so if we want to judge which ontological framework is kind of to be preferred or should be adopted, how do we do that? So how do we know what science is really telling us about the world? And the problem, and that is something that Elena and the others also highlight, is that within science itself, obviously, or even within specific disciplines, we do have opposing worldviews that are floating around, they're implicitly assumed. And so once we have that, it's kind of the question, well then, which science will we look at? What is our preferred, like how do we weigh the different approaches that we can find within a science itself? So what is the ontology that science supports? And the proposal I want to make, I mean, it's just, I mean, yeah, that's the proposal I want to make, but it's the, my approach here would be that to find an answer, we have to look not just at theory, but also at practice. And we don't just have to look inside science, but also outside of science. So I'm proposing that a solution to these disputes cannot be found just within science. And I'm not saying that Elena and the others have been like narrowly focused on science, but some of the statements can be read that way. And I'm just trying to propose it will have to be a broader analysis. So to give an outline, first, I'm just going to say a few things about the GMO debate and how it's entangled with certain ontological assumptions. Just repeat what other people have said about the topic so far. And then I want to go into the case study, and that's from pig farming, and that's in particular the porcine reproductive and respiratory syndrome. And that's a disease that costs a lot of money and causes trouble to pig farming. And so people are debating, how should we deal with that? And within these debates, different ontological frameworks are being employed. And so that's what I want to look at. So I'll say some things about the symptoms and the etiology of the disease, and then I will zoom in on kind of the genetic engineering, the gene editing that is proposed as a solution, and kind of the assumptions that are built into this approach. And I think this approach shows quite nicely that certain assumptions work really well, that will make more sense when I come to it. Then I will switch and I will zoom out of that kind of narrow focus on gene editing and look at practices and viruses and show that an alternative framework comes to the fore. And in this interplay between frameworks, I want to kind of draw some conclusions about how we should approach such debates about ontological frameworks. And that will be in the conclusions. All right, so the first part. Obviously, the debate about genetically modified organisms has been highly charged since its early days. And the debate is often framed, I think, as this fight between kind of industry and scientists on one side, as kind of the pro-GMO crowd, and then kind of eco-warriors and other non-scientists as kind of the anti-GMO crowd. I mean, it's not obviously a sharp distinction. There are scientists who are critical of GMOs and all that, but it's often portrayed in that way. And you find that in particular in kind of the arguments by pro-GMO advocates, that kind of this narrative that GMOs have been proven to be safe, there's no risk to humans and the environment, they provide clear benefits like high yields, pest resistance, and many more. And then there's also this narrative of kind of it's a precise intervention, like we know what we're doing. We have these genes that we move around and we have characterized them, so we know what will come out more or less. So it kind of gives predictable results. So it's this narrative of control and precision. And once you have that narrative in place, obviously it really narrows down the whole debate that you're having. Like opposition is now based on emotion and dogma. It's like a gut reply response to the technology. And it's the pro-GMO advocates often claim it's not based on science or reason, so it's kind of an irrational response. And obviously all of these claims have been challenged by critics, and some have also made the effort to kind of dig out the ontological assumptions that go into these pro-GMO arguments. And there's a very interesting paper, I think, by Kathleen McAfee from 2003, and there she discusses kind of what goes into the pro-GMO stance, and she identifies kind of what she calls a simplistic vision of the nature of genes and organisms that underlies these arguments, and especially also its ideas of precision and control. The simplicity, like the simplifying step, is kind of what she identifies, what I'll call here, isolate the box model of the world. And it's really this idea that each entity that we might be dealing with is what it is independently of its context. It's just its own thing. And that's often cashed out in terms of having intrinsic properties. It's these internal properties that it just has, which define the nature of a thing. And that is what this is close to what philosophers often call a substance ontology, where substance is a technical term within philosophy. It doesn't mean like matter or a compound, it's a more technical issue. And applied to this particular case here in the GMOs, and this is what McAfee discusses, within this box view or isolationist view, genes are treated as unitary objects that have given and stable properties and that we can move around from A to B, and they will still behave according to their nature, or however you phrase that. And that obviously has methodological implications. If you assume that, you can study them in isolation, you can combine them, and you can kind of extrapolate what will happen and so on. McAfee says that this really does not match with what up-to-date molecular biology is telling us. So she's basically saying this is out of date. This is not current science. And obviously she's writing at a time when the Human Genome Project was just about finished, and there were new insights into how genomes work and how genes are expressed and so on. And so she says, I don't know if you can see the pointer, but she says that the precision that is there is an illusion. Or the precision that is claimed is an illusion. Now, this kind of obviously the negative part is tearing down what these people are saying, the pro-GMO advocates. So what's the positive part? What is the alternative? And she's not very explicit on this, but Fern Wixon, who's also a part of this workshop, she had this book chapter in Edited Book, where she discusses this, and she discusses how opposition to emerging life technosciences, so including genetic engineering, is often based on the adoption of a relational ontology. And she uses the example of the deep ecology movement that began in the 1970s, roughly. And so in a relational ontology, the things that we're dealing with are not seen as isolated units floating in some sort of environment. Rather, any relation between an object A and an object B or a context B are part of what defines these entities. So the relations have a much deeper role to play. And she has a very nice section here. I'm going to cite, she writes, for deep ecology and its relational ontology, we do not exist as isolated units surrounded by a separate and external environment. Rather, we exist within networks of interrelation, engaging with various entities in a dance of co-creation. I think that's a very nice turn of phrase there. And so you can see it's kind of this focus on co-creation, there's also co-production, collaboration, interrelation. These are kind of key concepts in this relational account. McAfee uses that as well. And this whole talk of relational ontology is also closely connected to process ontology. And you have like current day philosophers who defend process ontology, and that's John Dupre, for instance, at University of Exeter. And I was part of his research project, so I would probably count myself as a process ontologist as well. But it goes further back. I mean, I just cite here Birch and Cobb, because they also wrote on ecosystems and philosophy of biology, but it goes back to Whitehead and even further back than that. So it's an old position, but it's closely related key elements to this relational ontology. Now, just quickly, I mean, what changes when we have this relational perspective? Obviously, context deeply matters now, and that has practical consequences for technology development, for instance. So if the gene is no longer this isolated thing that can just be moved around, any kind of fix is not as easy anymore because things can go wrong. It will not just behave predictably and so on. So it becomes more messy. But it's also the consequences for technology assessment, for instance, that matter. So that's something that Elena and Frederick discussed, that more data will have to be collected when you create a new context, new combinations of genes or a new organism, because you can't just extrapolate from what you've measured before. You might have to use different methods. So your whole mental framework of what you have to use and do changes. The assessment itself becomes more multidimensional because socioeconomic factors might, for instance, become relevant. And I think the pig farming example will show that. And once you have that, it's also that other stakeholders need to be involved. It's no longer just something that scientists decide. And many people who write on technology assessment have highlighted this, that it needs to be more inclusive and broader. Brian Wynn has written a lot on that as well. And so just generally, I mean, once we switch to the relational perspective, the way we think about science and technology fundamentally changes, you could argue. Now, what has happened so far now is that we've identified these two different worldviews at work. So there's this box model and the relational account. And so that was the step one in the solution that was proposed. But obviously the next step is now, so which position or approach is more convincing, better supported? How do we figure that out? Is it really all just relational, or is there some boxiness about the world? And it's a tricky question that I personally struggled with a lot. And some people would, for instance, turn to physics, and process philosophers do that a lot, and they turn to quantum mechanics, where it's all kind of fields and waves, and there's no real kind of isolated particles bumping into each other and all that. So you don't have a kind of a atomism anymore at that level. And this has influenced a lot of process philosophers, Whitehead most famously. And it's for sure, it pushes you towards a relational account, but then again, you could argue, well, it's kind of a mixed picture. It's not like every physicist is a process ontologist, even explicitly or implicitly. So it's like, it's not really just a given that this leads you to a relational view. And there's also strong reductionism in here. Like, is physics really where we should look at? Is this like the theory of everything? So it's, you know, you could question that move. Most people who discuss GMOs directly, I mean, they look at biology itself. So we've seen McAfee, and she kind of talks about the use of outdated genetics. And current day process philosophers do the same. Like, and I've done that myself as well. So I have a chapter in this book here as well, which is, by the way, open access. So you can look at that online. And it's kind of the claim, we need to look at up-to-date biology. And if we do that, kind of what we're going to see is it's all really relational. And so I've done that as well. I always struggled with that because at the same time, up to this day, scientists, and in particular molecular biologists and geneticists, they often do stick to the box model. And there are many cases where it actually works really well. And the pig example, again, will highlight that. And kind of the question for me is like, do they all misunderstand the very science they're working with? Like, for 20 or 40 years or more, they just have learned nothing. They just kind of are stuck in this weird way of thinking. And so that really raises for me the question that I mentioned at the beginning: on what basis do we identify the correct ontology that science presents us with? What science do we look at? Do we ignore the standard approaches in genetics, or which one, if they're different ones, how do we value them? And so this is where I want to turn now to the case study that I want to discuss, this porcine reproductive and respiratory syndrome. Just quickly first about the disease itself. It first appeared in North America in the late 1980s. Shortly after that in Europe, it spread to Asia and across the globe. So since the 80s, this has been pandemic, basically. It's a pig-specific disease. And the symptoms that you observe are reproductive failure, so you get abortions, premature births, and so on. Those piglets that are born alive, they have an increased death rate, or they have a reduced growth. And there are also respiratory issues in adults or in piglets, and that goes all the way up to pneumonia. And obviously, once you have that, it's a huge cost for the pig industry. In the US alone, it's over $600 million per year. And worldwide, this is several billion US dollars that is being lost due to this disease. So the pig industry is very actively interested in finding a solution to this. A key issue is that vaccines and drugs don't really work yet. So that's the basic. The disease is caused by an RNA virus. So you see a depiction over here. In orange, you have the a single-stranded RNA genome. You have some capsid proteins around it. And then you have here in light blue a lipid envelope. It's very similar. Basic structure is very similar to the coronavirus that's circulating at the moment. It's not a coronavirus, but the basics are very similar. This particular virus was identified in 1991, and we know that there are two genotypes. There's the type 1, which is mainly European. Type 2 is the US one, so they were identified in these different locations. And they're quite different. So they only have about 60% similarity at genetic level, which is not that much. Since 2006, there's also a new strain. It's called highly pathogenic PRRSV, which has appeared in China. So it's continuing to develop. And as I said, the disease is pig-specific, and it's even more specific than that. The virus mainly attacks macrophage cells within pigs. And so it does so by binding to surface receptors on macrophages using its own surface proteins. And that is really where the debate about how to treat this within genetic engineering has focused. So here you see a schematic of a pig macrophage and 5 proteins that there are more proteins in the surface, but these are the five of those that the virus seems to depend on in some way or other. And a key protein here has been identified as CD163. And this kind of molecular analysis combined with genetic engineering provides a very clean and clear narrative and methodology now. So the way people proceed in this case is you identify your problem, which is the viral infection. you identify host genes that are necessary for viral infection. So it has been shown that this is somehow involved in the virus uptake, so an infection itself. And then what you can do is you remove this through gene editing. And this is important because with the new technologies of editing, what you can do is you can just cut out this one gene. You don't need to move around genes. You don't need to introduce foreign DNA, some sort of toxin that you would use in pest-resistant plants. You don't even need antibiotic-resistant markers. It's literally just cut out that one gene, and that's all you do. So it has this very clean and precise narrative again. Then you test for effect on infection. Is it still happening or not? You can assess potential side effects on pig health, because obviously you're removing something that the pig normally has present. So is there anything going wrong for the pig? And then you get regulatory approval and you sell your modified pigs. So it's you can see it's a very attractive narrative for a commercial entity. It's like, let's do that, clear steps. And people have obviously started doing that. So this is a study from 2016. I think that was the first one. And what they did is they knocked out both copies of CD163 in pigs. And then they challenged these pigs with the virus in a controlled setting. And here you see, these are on the top bit A and B here, these are wild type pigs. And these are respiratory symptoms. And over here, you have pigs that develop fever. And you see after 7 days, the majority of the pigs have symptoms. Whereas down here, you have the knockout pigs, and there's just nothing happening. There's no fever, there are no respiratory issues that come up. So very efficient, very clean. There have been several other studies published in quick succession. These are just a few examples. You can see from the names, maybe, it kind of started in US and UK. And then there was a strong cohort of studies from China. So there's a whole global trade competition as well going on in the background that I will not have time to get into, but it's an interesting further dynamic here. Anyway, so all these studies kind of showed similar effects. Sometimes it's less clear cut than it seems here, but more or less it has been shown you can remove this and infection stops. There are some side effects of removing CD163 because obviously it has different functions within the pig, but by and large, the pigs develop normally. It's not like they have like 5 heads and 12 legs. They're more or less okay, I would say. What is very interesting as well is that you can actually take the pig CD163 and you can move it as a cassette to human cell lines, which are normally not infected by PRRSV, because it's pig specific, and you can express this block of DNA, this gene, or open reading frame, and then the human cells become susceptible to the virus. So it's really, I mean, I think it's an example that very powerfully shows that, you know, the box model does work every now and then, and it seems to be working here quite well. So what are critics saying? So we're talking about genetic engineering. Are there again these two poles fighting it out? This particular case, I think, is in very early stages. So it's just the last 3 to 4 years. It's, as far as I'm aware, it's not been approved by any government. Some companies have moved, some UK companies have moved to China, working together with Chinese state companies to get approval and get it to market, because China has a big pig farming industry. But in general, there has been relatively little attention and criticism so far, I think. There has been some tabloid headlines, you know, the Daily Mail, your usual suspects talking about scientists creating Frankenstein and all that. One critical voice I found was Helen Browning from the Soil Association. That's an association that does organic certifications, and they campaign for different issues, including against the use of GM crops. And so she said, we still know comparatively little about gene editing. DNA has a purpose at a specific time, and the process of selecting genetic structures and making changes could have unintended consequences. And then she goes on and she adds a little bit later, instead of putting on a sticking plaster, which for her is gene editing, we should be addressing the problems of our pig systems and looking to get the basics right. So, I mean, there's not much information in here. It's kind of a vague reference to unintended consequences and some systemic issues in pig farming. But, you know, a GMO advocate could say, or a genetic engineering advocate could say, well, does this really invalidate our approach? Because clearly the simplistic vision of genes and machines seems to be working quite well. there might be consequences that we don't intend, but they could be argued that we monitor them in test herds because we could just keep them in one facility and we look at what happens to them over years, or 1 year, they don't live that long. Because they get slaughtered. Genome editing, and you could argue that genome editing is up-to-date science. I mean, it's based on our latest understanding of how we can interfere with the genome and how it works. So, you know, I think people could make the case that the pig case shows that maybe the relational approach is kind of out of date and not staying up to date with the current science. So this is where I think it's interesting to switch to kind of a focus on practice and viruses. And I think that's the interesting bit about this example. When we look at I mean, there's two aspects that stood out to me when I looked at these papers by geneticists about pigs and genome editing. The first was that the dynamic, I mean, they talked about viruses, but they talked about viruses as kind of just the particle with certain genomic subtypes, and they bind to this protein or that. The dynamic nature of viruses itself did not get discussed in any detail in these papers. A second issue is that, I mean, the virus has been around for a bit over 30 years, or we've known about it for a bit over 30 years. And obviously people have tried to tackle this virus and have developed practices to kind of deal with it. So there's a whole complex of tried and tested methods that is being proposed. And this just doesn't get discussed. So usually these papers by the geneticists, they are, well, there's the disease. It costs the pig industry a lot of money. Vaccines don't work. That's usually what they mention, usually 1 or 2 sentences, they don't work. And then they go into discussing, well, there's CD163, we knock it out, we make the pig immune. And obviously that means that a whole section of work by virologists and practitioners in veterinary research is just completely ignored in these papers. So what I want to do in this last part of the talk is kind of dig into this part of the debate. And I think that shows that will point to a much more relational take. And I think we can learn from that. So when we look at virology and how it approaches viruses like PRRSV, it's intriguing that many virologists, not all, but many virologists now treat these not as a kind of a collection of 2 well-defined strains, type 1, type 2, and then, you know, a couple of subtypes. Rather, what they talk about is a concept they call quasi-species, or they talk about a spectrum of mutants. And the reason they talk about that is because RNA viruses, and PRRSV is an RNA virus, they have a very high mutation rate. So literally every newly produced viral particle contains on average 1 to 3 changes in its genome. Like it's a bit hard to measure it exactly, but that's kind of the current estimates depending on genome size and all that. But that's kind of the rough estimate. So in a sense, no virus creates exact copies of itself. It's more that the life cycle of the virus is more like a spiral. You never get out what you put into the cycle because there's always these little changes. So even within a single cell, what is reproduced is what these virologists call a mutant swarm or a mutant cloud. And that already tells you it's a very different take on what a virus is. And they make that explicit. So you have here a quote from 2 of so he's one researcher who kind of coined these terms. And they say quasi-species theory has encouraged viewing and examining viruses as complex mutant spectra and not merely as simple genetic entities that can be described with a defined nucleotide sequence, as believed only a few decades ago. So you can already see here the switch. It's not just like this complex of genes. It's a different type of entity. And what is really interesting in these debates about mutant swarms is that it highlights, once you focus on the swarm nature, they adopt the tools to kind of capture this as a highly dynamic entity. So what they find is that there are interactions, for instance, between different mutants. And these interactions itself, they can determine the reproductive success of the swarm as a whole. So this here would be kind of a swarm. Each of these blue dots would be a different virus. So they just run out of different symbols. So each of these has slight little changes. And they all interact. They're like cooperative interactions. They're competitive interactions. And out of this network of interactions, this is where kind of a different reproductive success comes about. And so the swarm can be more successful than just a single virus, or also less successful. a second thing here is that this dynamicity of the swarm is not just an intrinsic feature. It's not just that PRRSV has its dynamicity and measles has its and corona has its dynamic. The way this swarm behaves is highly co-determined. It's again it's again a dance of co-creation. You have a co-determination by the cellular environment within which the virus is developing or reproducing. And by that, also the environment of the cell, because the environment of the cell chain affects its interior and so on. So you've got this really long list of relations that comes out. I think once you adopt this kind of swarm view, there are a few important challenges that come up for the gene-focused approach to the specific editing case. For instance, the dynamicity of the virus would suggest that receptor switches could happen. And we know of cases where viruses have switched their receptors, literally from, not just from, I mean, this happened with the coronavirus right now, jumping from what animal bats and something in between to humans, which are, you know, same type of protein-ish, but very different. So you switch receptors, but you can even switch to using completely different surface proteins. And you could say, well, actually the PRRSV could, well, the mutant cloud could adapt to CD163 cells. There might be escape mutants in there that allow it to act differently. So if you approach this with the box model of genes, you could argue this is too limited to factor in this kind of dynamicity that viruses display, and that gene editing is really just a short-term fix. And obviously that's also a discussion with BT plants and all that. So I think this is already, I mean, this is again already a strong case for a more relational understanding, but you could argue, well, yeah, it might be short term, but it surely is a fix. I mean, you've just seen the data. It's like it really knocks it out. And yes, if you wait for a couple of years, things change. But like right now, right here, these genes behave like modules and all that. And also people might highlight that it's really, it's not every single, again, like with the physicist, it's not like every virologist is like, yeah, it's a cloud. It's like there are many virologists who talk about viruses as molecular machines. They just look at the particle and so on. So again, the question will come up, well, yeah, but like which virologists do we listen to? And I think here it's important to look at a second challenge that comes out. So this context dependence of viruses, I think together with the history of farming practices, suggests that the virus itself is a co-produced entity or a co-produced process. Again, this dance of co-creation. I think that's a very fascinating aspect of this case study. And so it's not just virologists who kind of tend to a more dynamic relational picture, but it's also veterinarians and also just the farming process itself that reveals a dynamic and relational picture. And I'm just in the last 2 slides, I think I'm gonna zoom in on this quickly. So the idea here is, okay, we have this dynamicity, the development, this means the development can be co-defined by the context of the virus, and that means farming practices might be part of what's going on. And there's a very interesting correlation here. So the emergence of PRRS, which is here in 1987-ish, coincides with big changes in the farming industry in the US. So here in gray, you have the decline of the number of pig farms. And in black, you have the increase in pigs per farm. So you get fewer farms, but they're larger. And this clearly coincides, I mean, in the 80s, this thing became pandemic. So it happens really at an important juncture in the development of pig farming. And the important bit is it's not just the size of the farms. It's not just, yeah, like bigger farms suddenly, but it's the dynamics and the organization of the farms. And so this is it's an interesting paper by Murto et al. These are from veterinary research institutes, and they looked at modern pig farming in the US, and they identified 3 key changes that happened. That there were many more, but these are the 3 key ones that they focus on. The first is that these larger herds that we now have were moved inside, so you have a more dense environment. They also highlight the invention of early weaning, so that means after 3 or 5 weeks after birth, you move the piglets away from the mother and you move them to a nursery. And importantly, there are also now different farms. So this is not just within a farm, but you have different farms that specialize on different parts of the process. So you have farms that just take young pigs and raise them to slaughter weight and so on. So you got movement between farms as well. And then you also have artificial insemination, which became a new norm. And what that means is like the virus has a completely new ecosystem and completely new dynamics, new ways of traveling within which it lives. Yeah. And that matters, especially the high density, high turnover context, because for a virus, that means it can develop into a more deadly or serious variant, because the virus usually tries to manage its level of lethality, because you don't want to kill your host too quickly. You want it to live long enough to bump into another susceptible target. But when you're in this high density environment, you can rip through the population, because there's always someone there to catch the virus. And you have high turnovers, you have a lot of susceptible animals and so on. So that really affects how the virus behaves. And Murto et al., they conclude, or they suggest that these changes suggest that the appearance of PRRS as a new disease of swine was due to ecological and environmental factors associated with changes in swine husbandry, rather than intrinsic changes in swine physiology and genetics. So you can see again this kind of shift in thinking away from like intrinsic things like this is the genome and that's just susceptible and there was this gene change and now it fits the pig and it came from somewhere else, blah, blah, blah. No, it's really kind of how husbandry has changed that gave rise. So it's really farming a new virus. And they conclude that perhaps the most significant long-term impact of PRRSV diversity and evolution will come from management changes to prevent PRRS. And so they're talking here about how we organize our farming practices. And so that brings up the idea of, you could farm a less dangerous PRRSV. And it brings us right back to these criticisms by Helen Browning and this focus on practice of pig farming and getting the basics right. So now we see kind of this idea that we can create a stable and safe situation by adjusting farming processes. And it could even lead to the elimination of PRRSV altogether. And that has been done. So there are examples where you would use herd closures. So you don't bring in new animals for a long time of days. You combine this with vaccination and how you change animal handling and animal flow and biosecurity measures. And with this block of measures, they could reach the local, achieve a local elimination of the virus. One study in Minnesota, one in Denmark, and that's like across like 12 different farms, like a secluded area where they could completely eliminate it. And I mean, obviously the approach needs a lot of coordination between different players. It's costly, it takes time. So it's not, it's not the industry's favorite approach. But it's important here, this is not a molecular approach at all. It's really about process adjustment. So it's really a fundamentally relational way of approaching the issue. So I think what these different visions show is that once we factor in findings from virology and veterinary research, and if we put all of this in the context of actual farming practices, suddenly a relational worldview seems to be kind of quite well supported or better supported, makes more sense maybe. So that is kind of why I'm thinking through this example made me think it's probably, when we just focus on science, we're always caught in this, well, but other scientists are saying this. Or say, once we open this and look at different sciences, for sure, genetics, virology, veterinary research, and we also look at outside practices, the larger way how things work, so to say, this can give us a quite clear verdict on which ontological framework is more reasonable. And this has been 38 minutes, and so I come to my conclusions. Just quickly summarizing again, I mean, as many people have shown, there are competing ontological frameworks mobilized in these discussions about genetic engineering. I want to claim that the case of genome editing in pigs is a quite powerful support for box model at first sight. That doesn't seem you need a relational view. But what I hope the case shows is that once you bring in the larger context and in particular focus on practices, including farming practices, the box model and the gene editing that feeds on it, or the gene editing approach that feeds on it, this clean narrative, starts to lose its appeal. And so I think it's important that we not just think virus particle or a virus plus environment, but really kind of a virus in practice. And once we switch to this practice-focused approach, a relational view starts to emerge as a more complete and reasonable view. And so that brings me back to the initial question, where I want to propose that the focus on ontology assessment has to be both on science, its theory and practice, and on broader practices outside of science. And that's all I have. And that's a thank you slide. And I have, just in case anyone wants to look at all these citations I mentioned. And thank you for listening, and I'm looking forward to comments.